Phytocannabinoids produced by the cannabis sativa plant have been used both medicinally and recreationally for thousands of years. More recently, interest in the medical use of CBD, THC, and other phytocannabinoids has exploded. Changes in the legal status of the plant around the globe have spurred intense research interest into the pharmacology of these compounds. Exactly how do these molecules work their magic in the body? The answer to this question involves a detailed explanation of the endocannabinoid system, or more simply, the ECS. The ECS is thought to be 600 million years old. It is present in every animal species, except insects, and has evolved as an extensive endogenous signaling system, which allows cells to communicate with each other so that balance is restored following a cellular stressor. Examples of cellular stressors include infections, injuries, acute and chronic disease, and more. There are three primary elements of the endocannabinoid system: cannabinoid receptors, the endocannabinoids themselves, and the enzymes which both produce and degrade them. Cannabinoid receptors are G-protein coupled receptors. Two of them have been identified to date, CB1 and CB2. The CB1 receptor is the most densely populated G-protein coupled receptor in the human central nervous system. CB1 receptors are found on many cells throughout the body, mostly located on nerve cells or neurons and glial cells in the central and peripheral nervous system. CB2 receptors, on the other hand, are mostly located in the periphery, in organ tissues, and on immune cells. Endocannabinoids are lipid signaling molecules. Arachidonic acid is the principal omega-6 fatty acid that is used to produce endocannabinoids. In the central nervous system, these compounds act as fatty acid neurotransmitters, which are synthesized and hydrolyzed rapidly. They have very short half-lives in the body. Two endocannabinoids have been isolated and characterized to date: arachidonyl ethanolamine, or AEA, also known as anandamide. Ananda is taken from the Sanskrit word ananda, which means bliss. The second is 2-arachidonyl glycerol, or 2-AG. A quick clarification before we proceed. The prefix endo in the term endocannabinoid refers to the source. Endocannabinoids are produced endogenously, meaning inside the body. Phytocannabinoids, on the other hand, are produced by the plant. The prefix phyto suggests that their source is a plant. Synthetic cannabinoids are a third category. They are synthetically produced and exist primarily as pharmaceuticals or illegal street drugs. We won't talk much about synthetic cannabinoids in this video. Notice how different endo and phytocannabinoids appear from one another in terms of their chemical structure. The discovery of the endocannabinoid system began with Israeli researcher Raphael Mechoulam and his team at Hebrew University in Jerusalem. They set out to find the active ingredient in cannabis, or marijuana. CBD was isolated from cannabis in 1940, but it wasn't characterized structurally until 1963, more than 20 years after it was first isolated. Initially, CBD was deemed an inactive constituent because it failed to mimic the effects of cannabis extracts when administered to animals and humans. As a result, CBD was put on the shelf. THC was isolated and characterized in 1964. It did mimic the effects of cannabis when administered to animals and humans. Consequently, THC was deemed the principal psychoactive ingredient. With this information, scientists wondered how THC worked in the body. Using the discovery of the endogenous opioid system as a blueprint, they hypothesized that a receptor in the brain must be involved. In 1988, researchers at St. Louis University discovered a receptor that was activated by THC. They named it cannabinoid receptor 1, or CB1. This discovery raised an important question. Why do humans have a receptor for a molecule whose only natural source is a plant? It was hypothesized that we must make our own version of THC. In 1992, scientists discovered the first endocannabinoid, arachidonyl ethanolamine, or AEA, or anandamide. In subsequent years, researchers identified a second cannabinoid receptor, CB2, and a second endocannabinoid, 2-arachidonylglycerol, or 2-AG. If it's not already clear, endocannabinoids bind to cannabinoid receptors. The interaction between a molecule and a receptor is often described like a key fitting into a lock. When anandamide, for example, binds to the CB1 receptor, it activates the receptor. This typically results in something happening inside the cell. That something is typically a biochemical cascade that leads to a variety of downstream effects, like a gene being turned on, for example. Anandamide binds to the active site on the receptor. When it's the CB1 receptor that is activated, that something could be an effect that we associate with cannabis. To be more specific, activation of the CB1 receptor could result in euphoria, or pain reduction, or appetite stimulation, or nausea reduction, or any number of effects that we associate with cannabis use. The concentration and location of these receptors typically determines the observed effects. High concentrations of the CB1 receptor have been observed in the cortex, the basal ganglia, cerebellum, hippocampus, and other areas of the brain. CBD binds to a different site on the receptor. CBD doesn't activate cannabinoid receptors. Endocannabinoids are signaling molecules. Like hormones and neurotransmitters, cells use them to communicate with one another. In the nervous system, endocannabinoids act as neurotransmitters. Dopamine, serotonin, and glutamate are other neurotransmitters you may have heard of. Nerve cells, or neurons, use neurotransmitters to communicate with one another and with other cells outside of the nervous system. Dopamine, serotonin, and glutamate are peptides. Peptides are proteins. Endocannabinoids are fatty acids. Fatty acids are lipids. This is one of the ways that endocannabinoids are different from other neurotransmitters. In this diagram, there are two nerve cells, or neurons, a presynaptic neuron and a postsynaptic neuron. And then there is the synapse that occurs between them. In contrast to classic neurotransmitters, like glutamate, for example, which are synthesized ahead of time, stored in vesicles, and diffused from the presynaptic neuron to the postsynaptic neuron, endocannabinoids are synthesized on demand and diffuse in the opposite direction. They bind to CB1 receptors on the presynaptic neuron. This causes the synaptic vesicles spilling glutamate into the synapse to retract. In this model, endocannabinoids act like a shutoff switch, protecting the postsynaptic neuron from injury due to excessive excitation or excitotoxicity. Endocannabinoids are not limited to cellular communication inside the nervous system, however. The ECS communicates with all other systems in the body. Here is an example of immune regulation. The preceding example was an example of neuroregulation. In this particular example, we're going to look at anti-inflammation. This is a diagram of inflammation in the gut, where white blood cells like mast cells and macrophages have infiltrated the wall of the gastrointestinal tract. Inflammation ensues as these cells produce and secrete inflammatory mediators like TNF-alpha. Anandamide is also released as an anti-inflammatory. Anandamide diffuses and activates CB1 and CB2 receptors on the macrophage itself. This causes a decrease in the secretion of TNF-alpha and other inflammatory mediators. It also diffuses to a nearby mast cell and binds to cannabinoid receptors there. This leads to the same effect. CBD works a little differently. As mentioned earlier, CBD does not activate cannabinoid receptors, so the mechanisms we have reviewed so far do not pertain to CBD. Scientists are still trying to figure out exactly how CBD does what it does in the body. We do know a few things, however. While CBD does not activate CB1 or CB2 receptors, it does activate other receptors, non-cannabinoid receptors. These receptors are outside the ECS, scientifically speaking. For this reason, CBD has been called a promiscuous molecule. Over 65 molecular targets have been identified to date. Some of these targets are receptors, but others are enzymes or transport proteins. For example, CBD slows down the enzyme that breaks down anandamide. This enzyme is called fatty acid amide hydrolase, or FA. By inhibiting FA, levels of anandamide are elevated and stay that way for a longer period of time. This may be one of the key mechanisms underlying CBD's therapeutic effects. Another example involves a serotonin receptor. There are several serotonin receptors. CBD activates one of them. The serotonin 5-HT1A receptor is a known drug target for anxiety and nausea. Perhaps this is how CBD reduces anxiety and reduces nausea and vomiting. CBD also activates the capsaicin receptor. This receptor is named after capsaicin, the active ingredient in chili peppers. The capsaicin receptor is associated with pain and inflammation. Perhaps this is how CBD exerts its anti-inflammatory and pain-relieving effects. It has been suggested that dysregulation or alterations of the endocannabinoid system may be associated with various health conditions. Ethan Rousseau, an author and researcher in the field, has categorized deficiencies in the endocannabinoid system as clinical endocannabinoid deficiency. Research shows that an underlying endocannabinoid deficiency may be involved in the pathophysiology of multiple conditions, including Huntington's disease, migraines, multiple sclerosis, IBS, fibromyalgia, and others. In closing, our current understanding of the endocannabinoid system is largely based on animal models, and it is still evolving. While there are many possible therapeutic applications, further clinical trials are warranted in order to improve our understanding.